Welcome back, everyone. I hope you had a really nice weekend. We'll kick off this morning and this week with Brunton, who will talk about neural decoding in the wild. Get away, Rick.
Thank you, Ariel. Thank you, Noah. Hi. Just give me 1 second here. Yeah, hi, my name is Bing. For those of you who don't know me, I'm here at the University of Washington. I primarily sit in the Department of Biology, and I also have some relationships with the eScience Institute for Data Science, Computer Science, Applied Math, and couple of methods-driven departments. And I also take graduate students from the neuroscience graduate program. And so I'm really at that interface between biology and neuroscience and data and behavior and neuroscience of all kinds. I do methods development primarily so that I get to work on all kinds of really cool things. And I'll try to focus on only one of them here today. I did get an announcement through my phone this morning, the first thing I woke up, from the University of Washington saying that they may be cutting our power and air today. And I was like, darn it, because my friend who works at Google just sent me a picture of Friday, and he's like, look what I got at work. And it was a matcha latte. It's like, great, you get matcha latte at work. I don't even get electricity and air. But it is. Anyway, so I think we're going to be okay. So far, the power is on. It's reasonably cool in here. I think we're going to make it. If we don't make it, we're just going to go outside. All right, and I'll finish the rest of my talk outside. It's going to be great. Awesome. You ready? All right. Oh, like I said, you want me to do 60 minutes? Yes. 60 minutes, because there are clock in here. There is that clock in there.
That clock is there to confuse you.
Yes.
We can give you 10 minutes away.
Okay. I'll just get my phone. The reason is because I'm not gonna I don't want to talk the entire time. I very much actively encourage everyone to just interrupt me and just yell at me if you have questions. I'll try to stop in the middle if there's a good place to stop. But otherwise, just feel free to raise your hands. Folks on Zoom, say something. Maybe somebody else, could one of you, Noah or Ariel, just yell at me if somebody has a question. Otherwise, I don't know if you're hooked up to the speakers in this room or something. You might sound like a deity coming from the sky if you ask a question, but that's totally acceptable. I will stop talking if you copy. So I'll just end at 60 minutes, right? And if I don't get to the end of my slides, it's totally cool. That sound good? You got a deal? All right, I'm gonna yell at you and you can yell at me. So this is gonna be very mutual today. Yeah? All right, cool. So before I get started about actually the thing that I wanted to talk about today. Interesting. All right. Maybe. I wanted to tell you a little bit more about what the rest of my lab works on. So as a computational neuroscientist, one of the things that's really cool about my job is that I get to collaborate with a bunch of people. And so since I'm not actually responsible for collecting data or doing any experiments, I get to work on all kinds of really cool organisms. And so we work on everything between insects and humans. There's a really cool bird project going on. We work on some rodent stuff. I've had some work on non-human primates in the past. And currently, there's actually a ton of work on insect neuroscience. That's been really interesting. What ties it all together in my mind is that I primarily feel like I work on data-intensive methods to understand your computations, especially underlying natural behaviors. So brains doing whatever it is that we brains like to do in the wild, right? The thing for which we evolved to do, which is not to sit on do. But we work on everything between insects and humans. There's a picture of my daughter Abigail on the right-hand side in her natural habitat doing what she likes to do best, which is eating spaghetti. I don't actually study her, but I needed a picture of a human, so up she went. In my spare time, I've also been doing some painting. So one of the things that I decided is that I picked up watercolor painting, and I realized that there was just a ton of people doing watercolor painting of landscapes. As much as I like landscapes, the world did not need another person painting landscapes. The world needed somebody else to paint obscure insects nobody paints. And so I picked up I call them glamour portraits of insects as well. So here's a couple that I painted in the past. Sometimes I paint experimental organisms that my friends work on. And so that's been really interesting. Okay, cool. So on to the topic of the actual talk today, we're talking about brains. We're specifically talking about human brains, which is a type of mammalian brain. So there's a couple of pictures here of mammalian brains, and you can see that they have, they're really different, but they also have a lot in common as well. Some of them are wrinkly, some of them are not. On the right-hand side here is a human brain. To the best of our knowledge, it has approximately 86 billion neurons. And each neuron is connected to another neuron, approximately 1,000 to 10,000 neurons. Okay, that's a lot of connections. This is a really enormously complicated thing that we are studying here. So one thing that I like to do when I'm first studying this, you've probably seen this before, is if you put your two fists together and take two fists and put them together and you look at it, that is approximately the size of your brain. Now, the variance in size of human brains is significantly smaller than the variance in size of human hands. So it's my hands are relatively small, so I think my brain is quite a bit larger than this. Some of you may have larger hands. It doesn't mean anything. All right, cool. So brains are cool to look at. They're like anatomically interesting, but there's a lot more to them that make them also really interesting to study, especially for someone like ourselves. So brains have multi-scale structures in space. What does this mean? What we're looking at on the left-hand side are logarithmic coordinates of size scale. So every tick mark as we go up is 10 times larger. All right. So the smallest size scale down here, something that's especially of interest when thinking about neurocomputation or neurofunction, is a single synapse, a single connection between one neuron and another neuron. We talked about 86 billion neurons in human brain. 10,000 ish synapses per neuron. So there's like exactly a gazillion of these things in your brain right now. Okay. Now, here is a small picture of all of the proteins in a synapse that I pulled up for a review paper. And the thing to note here is that despite in computation, we often simplify the synapse as a single number, like sometimes a linear weight. It's just like a number, it's a weight, right? Maybe if I felt like that, I slap a nonlinearity on top of it. But in reality, the biophysics and biochemistry of a single synapse is enormously complicated. There are whole conferences of people where they get together, hundreds of thousands of people just talking about a synapse. It's so complicated that if you look at the protein structures that hold the whole thing together, the plasticity of it, the ways that in which it regulates itself, it has the complexity of a small city. Not a huge city like London and Manhattan, and but like more than a one-stoplight town, right? It's got garbage service, it's got highways, it's got recycling plants, really green, so everything gets recycled, nothing gets wasted. And so that's one synapse. And you go up in complexity scale of dendrites and neurons and layers of stuff and nuclei and lobes and up to a brain and up to the order of a meter, which is the size of a human body, right? There are interesting things happening at every single one of those size scales. It is not obvious where you should cut it off and make an approximation and say, okay, this is the correct size scale with which I should be describing the function of a human brain. It's really all of these things, right? Super complicated, but also that makes it really, really interesting as well. But there's more, because not only does the brain have multi-scale structures in space, it also has multi-scale dynamics in time. So at every single one of those size scales, there are interesting dynamics happening and things that change in time. And that gets me really excited because one of the reasons that I got into neuroscience in the first place is because I love dynamical systems so much, the mathematics of things that change in time, right? If things don't change in time, well, anyway, I guess I can go on down anyway. But so here we're looking at time scales on the horizontal axis, where once again, every tick mark is one order of magnitude longer in time. Now, the shortest time scale that we often think about, at least in systems neuroscience, is 1 millisecond, because that's about the duration of a single action potential. Okay. Anything shorter than that, you can actually keep going a couple of orders of magnitude if you wanted to think about protein chemistry and folding and protein kinetics. You can get down to at least femtoseconds if you wanted to think about protein dynamics like that. Okay, I didn't push it out because I didn't cut a diagram for it, but you can keep going unless you wanted to. And between the scale of milliseconds and decades, which is the duration of approximately human life, there is interesting stuff happening at every single one of these time scales that people study, right? We have things like synaptic plasticity and neuromodulation, which happen on the order of tens of hundreds of milliseconds. By the time you have sensation of movement and control of speech, you are talking about control over the course of tens of milliseconds. But of course, understanding requires short-term memory. I've been talking here for about 5 minutes now. And you've been integrating in your head what I said in the beginning when I started talking with what I'm saying now in order to form an understanding when you think I'm going, right? Of course, there's been things like decision-making and long-term decision-making. So decision-making is not just, you're rat in the maze, go left or go right. Decision-making is also like life decisions, like where to go for graduate school, who do you choose your life partner, where do you want to live next? Big decisions like that. All along, there's things like circadian rhythms, so seasonal hormonal fluctuations, and all of that adds up over the course of decades to long-term memory and learning. And that forms a picture of who you are at this moment as the accumulated stuff of what's been happening in your entire life. Okay. One interesting fact that I was really distressed to learn when I first learned about this. Remember we talked about the synapse and all the proteins that are holding your synapses together? Okay. If you look up, because proteins get recycled, right? Those of you who have maybe taken molecular biology or cell biology at some point, you remember that the proteins, they have a limited lifetime. They don't last forever. And so part of what your body does is why you keep eating all the time because you need extra energy, is to keep up all of the proteins that hold your body together, right? Your skin cells are constantly dying, you're swapping them off, you're making new ones. You're not making new neurons, not many anyway, but you are recycling all of the proteins in your body, including all of the proteins in your synapses. I looked up the half-life of your protein in the synapses in your protein. The proteins in your synapse. Thank you, Ariel, for the coffee. I needed it. Okay. The half-life of the proteins holding your synapses together is like days to weeks. That means every couple of weeks, every single one of the proteins holding your synapses together is being ripped out and replaced by a new one. Just think about that. What does that mean for who you are, right? So the story goes back to there's a story of Jason the Argonauts has a ship, right? Builds a ship, there's a ship, this is the ship. Okay. So one day, one of the planks on the ship breaks, and they're like, "Oh, we'll replace it with a new one." So they get another piece of wood, they nail it in, it's a new one. And then the railing breaks. So they're okay, fine, we'll get a new piece of railing, right? And over time, every single piece of wood that went into Jason's ship has been replaced by a new piece of wood. Is it still the same ship? Now, this may sound like a philosophical discussion, and it totally is, and we're talking about Jason's ship. Now we're talking about your brains. Right? Because that's true of every single protein in your synapses. Like your synapses have almost no proteins in common with whoever you are 10 years ago. Are you the same person? I have no answers. I'm just asking questions here. Actually, I'll pause here if anyone would like to venture a comment before we dive more into more technical stuff. All right. Think about that. We'll talk about it at lunch. Cool. So, how do we study all of this? This is like overwhelming complexity, right? We have these time scales, have these length scales. You can have a perfectly good career spending your entire life at the intersection of any of these intersections. Pick a length scale, pick a time scale. It's like, okay, this is me, I'm right here at the crosshair. I'm gonna make my career right here. I'm gonna spend my entire life trying to understand this thing at this time scale and this length scale. And that will be a perfectly fine career, right? Look around at workshops and conferences in neuroscience. There is one for almost every intersection of those time scales and length scales. But that's not what interests me, because if you look at it as a whole, it's pretty obvious to me that the interesting stuff is not at any particular intersection of size scale and time scale, but rather how all of it is integrated together. And so if you zoom way out, instead of trying to understand a single species or a single brain, you think about it in the context of evolution, it seems rather self-evident to me that the brain evolved to move the body. Not the human body per se, but any animal body. And in fact, this is what distinguishes us from plankton, right? We have control and agency over where we are in space and time. And that is one of the reasons, one of the fundamental reasons for which the nervous system evolved in the first place. And so if we acknowledge the fact that the brain doesn't exist for having philosophical thoughts about shipbuilding, but rather to move my body around, right? So like I'm an inverted pendulum. I'm intrinsically unstable, mechanically, not like, I'm standing here right now and I'm spectacularly succeeding in the feat of not falling over. That's difficult, computationally speaking, for me to just constantly not fall over at any moment, even though I'm also talking and waving my arms around, right? I'm gonna try to drink a coffee for a second. We'll see if I spill it on myself. So if you acknowledge the brain for behavior, that really changes your perspective, to change my perspective rather, on how to approach understanding the brain. So some of the approaches that we've been increasingly taking in my lab include things like studying natural behaviors rather than very well-controlled behaviors. I want to know what brains actually do, what they're good at doing, rather than the thing that which it manages to do after I overtrain it for all the time. And in addition, in the context of evolution, we've been taking increasingly comparative approaches, right? Thinking about the natural experiments that are out there in the natural world, looking at not just a model organism, but a set of related model organisms that are similar but different in interesting ways, and taking those comparative approaches as well. The second thing, like I said earlier, is acknowledging the fact that the brain evolved to move the body around, and the body and the brain evolved together. That means the body matters, right? You can't ignore the body. This is not an abstract brain in a jar situation. The body is there to work with the brain, which means the details of the biomechanics and the way in which the sensory apparatuses are distributed throughout your body and the way that your muscles are connected to your body actually matter as well, because that's the actual input and the actual output of the brain, right? The brain is connected to the outside world approximately not. It's only connected through all of your sensory perceptions and all of your muscles actuations. I'm exaggerating a little bit. Of course, there's interoception, you have internal organs, all that stuff, but that's what you know what I mean, approximately, right? So that's what the whole system is, right? This is almost like a systems engineering perspective on neuroscience. You have to actually think seriously about the entire plant, the plant here being the animal, and figure out what this means. So what this means for me in my research program is that we're increasingly thinking about morphology and mechanics. I don't think I don't think there's such a thing as passive perception of any kind. Like there's no just vision, right? Like vision isn't just looking at things. Vision is looking at things, deciding what I'm going to do in the next moment, and anything I do changes my vision input in the next second. Like anything I do, right? Saccades, moving your body around, making decisions about how I move around in the world, that all changes my sensory inputs in the next moment, even vision, which is typically studied traditionally in a passive context. So morphology and computation matters. And then we also think about embodied computations. Any computation that can be pushed out to the periphery, that can be done like locally on the body, like tactile sensation, for example. Anything like that would make the computation more efficient instead of having to have more demands onto the central processing unit, the central nervous system. And in addition, there's latency issues. Anything that has low latency and can be computed very quickly without spending the time to go from the end of my fingertip all the way up to my brain and back would be faster, right? Does anyone know so this is another neuroscience factoid. The longest cell in your body. What is the longest cell in your body right now? You all have one. Any guesses?
The longest, as in like,
Longest single continuous cell, a single cell that is the longest in your body.
in the vagus nerve.
Good guess, good guess. We can go longer. Go ahead.
I think there are motor neurons that go from the cortex to the base of the spinal cord.
Good, we can do a little bit better than that. Yeah, that's a good one. So you're already thinking about the sensory motor system, which is good. Keep going. Yeah. Ah, very good. But where's the other end? Brainstem. Yeah, it's the dorsal root ganglion. There is one that's in the very base of your spine, the last vertebrae, right before your tailbone. Okay. The cell body is in the dorsal root of the spinal cord there. One end of the cell goes to your tippy toes. The cell body is in your spine, okay? One end goes to your toes, the other end goes to your brainstem. So there's a cell in your body right now that is almost as tall as you are. The giraffe has one too. And so does a whale. And presumably, so did dinosaurs. Although paleo neuroscience is a little difficult, experimentally speaking. Like, if their nervous system is organized like every other vertebrate we know, there was a neuron inside one of those, like things that are like 10 times, 100 times taller than I am. It had a single neuron that presumably went from the very tip of its tail all the way up to its brainstem. Likely the longest cell that's ever existed. Right? That's a lot of wire. And there's a lot of wire there because feedback is hard. Right? Because the time it took to go from the very tip of my toe up to the brainstem, for my brain to decide, oh, that hurt, and to send commands back, actually takes too long. And so if it's possible to do it without feedback, I should, but if I have to plan forward, I should do feedback, right? Feedback is central to every single aspect of biology. This is not a neuroscience thing. Feedback is central because when I push on something, if I push on this table, it pushes back, right? If I talk to you and say something funny, you laugh. Yes, you do. Okay. Inside the body, neural circuits have feedback. There's tons of recurrence. Inside every single cell, there's a ton of feedback, right? There's transcription regulation, protein circuits. You don't have the answer right now, because I don't want to put you on the spot, but like I challenge you to name one thing in biology that does not involve feedback. Buy your coffee, anyone who names one. That genuinely does not involve feedback. Okay? Yet in modeling, most of us persist in modeling things like they're only feed-forward systems. No recurrence, no feedback. What madness. So I've done it. I'm going to do it for the rest of the talk, so don't worry about it. So this is just where we're going, right? A lot of what I've been doing in my lab is building integrated sensory motor models. So not just sensory models, like encoding models or decoding models, but all of the above, all of it together. And then we're also, of course, using reinforcement learning techniques to think about not only what the execution of the behavior is, but how do we actually execute goal-oriented behaviors? How do we actually accomplish something to get a reward that we actually want for survival? Okay. So this is kind of where we're going, but not what I'm actually talking about today. I just want to tell you that there's things we work in my lab. There's a ton of people working as far as sensing and control for both biological and engineered systems. There's a cool set of stories there. We've done more than a little bit of work on quantifying natural behaviors. So developing both computer vision as well as dynamical systems and analytic techniques to quantify natural behaviors, connecting natural behaviors to brain activity. Usually there's a couple of people in my lab just doing applied math of various kinds, building models of dynamical systems, sometimes with biological applications, sometimes not. And then the thing that I'm actually going to talk about for the rest of the time today is neural decoding and brain-computer interfaces, especially in uncontrolled scenarios. Okay? I just want to tell you, like, I wanted to take the opportunity that I'm here, standing in my little virtual soapbox, to tell you how I thought about things and why I'm a neuroscientist and the things that I think about. And we're going to spend the rest of the time talking about neural decoding and brain-computer interfaces. Is that good? Great. Awesome. I find myself doing that a lot more after spending so much time on Zoom, because I'm always muted. I'm like giving people thumbs up, and now in real life, I'm giving people thumbs up a lot more, right? It's like a really nice way to give feedback without verbally interrupting them. It's like, yes, I'm with you, right? It's like, makes me less dizzy than just nodding a lot. Okay, cool. Sounds great. Okay, so brain-computer interfacing. So this is something that it's a huge field, right? If I had to think about it, there's basically, for me, 3 layers of brain-computer interfacing, 3 reasons for why we would like to build computerized devices to interact with brains, especially human brains. The first one is to assist individuals with physical and neurological disabilities. Maybe they have a capacity that they would like to have or used to have, they no longer have for reasons that are clinically relevant, and they would like to be able to have those abilities again. The second reason is to augment engineered systems, humans in the loop. This is the kind of thing which is like, getting a little bit more sci-fi, a little more towards the future, right? There's capabilities that we would like to have that we don't quite have, and we can interact and collaborate with computer devices. This can be a really cool thing, right? So the most trivial use of this is like the trackpad and the keyboard, right? Non-invasive, but this is a brain-computer interface. I'm interfacing my brain with my computer using the muscles in my hands, right? And this gives me superpowers. I can look up like random factoids on the internet really fast. And this is the capability that I as a human did not have before that I now have because I can interface with my computers. And so if you can do that more seamlessly and without having to take your eyes off the road, for example, that would be a capability that would be an augmentation of current human capabilities. And the third one, which is even more sci-fi, is to enhance one's daily life in an increasingly data-rich world. We are living in an increasingly data-rich world. We all have these supercomputers in our pockets. I don't refer to this device per se, but the access that we have to computing through devices like this that we constantly have in our pockets at all times. So this is just a reality of the world. So there's a capacity for brain-computer interfaces to enhance our interactions and enhance our connections, not only with computers, but with each other. I think this has already made a huge difference in our lives. And so it's something that we should think seriously about. I'll point out that like all technologies that humans have ever invented, brain-computer interfacing has the capacity for doing good as well as evil. And so we should think pretty seriously about not only the tech side of it, like what can we do, but think seriously about it beforehand instead of only afterhand about what are the capacities for this to be abused by people who are intent to, because somebody will be intent to do so. Just acknowledging that, okay? All right, so two of the things that I have found the most impressive in recent memory, in the areas of brain-computer interfacing, is I'll do this one first. So this came out a couple of years ago, speech synthesis from neural decoding. So I remember the first time I saw this result. I think I saw it at a workshop. Gopali came and gave a talk before this was published, and the first time he like played this video of direct speech decoding from one of their brains, like everyone's jaw dropped, right? It was like absolutely astonishing. One of the coolest things I've seen in brain-computer interfacing in recent memory. And of course, these folks, as well as their collaborators, have done even more impressive things in the years since, but in the vein of direct speech decoding from brains. A bit earlier than that, and this story goes back to a little bit more like when we started this project, the brain-computer controller robotic arm. This is a group out of the University of Pittsburgh. Here we have Jan. Jan is our quadriplegic, and she hadn't been able to move either her arms or her legs for about 10 years before this procedure. What you're seeing here is, so she's got these two gray boxes up above her head, right? They each have about 100 electrodes connected to her motor cortex. And they developed a way to decode her brain activity to control this robotic arm. And so here is Jan feeding herself chocolate for the first time in 10 years. And she looked very happy. I would be too, if I hadn't been able to feed myself chocolate for 10 years. Okay. So this is super impressive. And so the story is going to start actually so we started working on this in 2014 with my first graduate student, Nancy Wong. Okay. Nancy and I saw this result. Again, we were like, oh, this is just so cool, right? We want to work on this. So we were looking at the set of results that was coming out from the team at Pittsburgh and a couple of others around the world who've been doing this ever since. And we identified three things that we thought we might be able to contribute to in this field. Because we weren't actually doing experiments, right? Like, I'm a computationalist, she was a computer scientist. And so we were looking at this picture, and here are the three things we identified that we thought we could contribute to. The first one is, how do we take this outside the lab, right? This is a whole big apparatus where Jan goes to the University of Pittsburgh. She sits there in a lab, right? And the apparatus only works inside the lab. And so for this to be useful for her, she has to be able to take it home somehow. So I'm like, I'm usually thinking about future technologies, right? Instead of I'm not like much of an engineer in the way that I solve actual problems that are in front of me. I think about future problems that would invariably exist in the future. So I'm thinking about the future about when people like her will want to take it home. What are the technological challenges that we should anticipate so that she can take it home later? The second one is, how do we automate this? This was Nancy's idea. There's a graduate student. There's in fact a team of technical people making this work for Dan every single day, right? This again is super cool, but it's not really scalable. And you can't take your own graduate student home so that you can feed yourself chocolate in the morning, right? So how do we automate the fact that this takes a huge amount of human effort, fine tuning all the parameters so that this would work, right? The secret behind all of these decoding results that are super impressive is that they need to be retuned every single day and they need to be calibrated. There's a whole like half-an-hour long calibration process before she's able to use a robotic arm every day. Like, again, sounds great, but that's not really useful if you're taking it home, right? And you certainly don't get your own dedicated team just sitting there like fiddling with the parameters and rerunning and recalibrating your models every day. There's a brain trust. This is not good. And the third thing we identified is that how can we build models that adapt to other people, right? A lot of these models are incredibly bespoke. They are tuned for that individual, which is great for them, but this is not a scalable solution, right? We can't afford to train special models for every single person. And also because it takes so much training data. When you had a new person who came in and needed something like this, what do we do? So over the last, I don't know, I remember since Nancy first started working on this, we've been tackling these questions more like at a conceptual level, right? Thinking about what are the algorithms that we need to develop so that we can make it that you can take these kinds of brain computer interfacing devices outside the lab so that we can automate the training and calibration process, and so that we can transfer the ultimate models to individuals with either minimal retuning of the models or with just having them be able to work sight unseen. This is like the vision of what we set out to do, and I'm going to tell you a little bit more about what we've done ever since. Okay, so first I'm going to talk to you about the data set, and this data set was really like I would say it's like a heroic data set because Nancy did this when she was a first-year graduate student, and she had to fight with ICOOK and not with ICOOK with with the IRB, which is really impressive for a first-year graduate student to push through that much paperwork at a hospital, right? Because what we realized was that there is a set of procedures that our friends at the neurosurgery department at Harborview Hospital downtown were doing for patients with intractable epilepsy, right? So has anyone talked about these clinical procedures? Okay, so I'll do super fast because you've heard about them before. The procedure is basically for folks with intractable epilepsy so that they can localize the source of the seizure and do a targeted resection surgery. Right? They're going to surgically remove the piece of tissue that's hopefully causing the seizure and alleviate the symptoms because nothing else is working for them. For us, this is a week-long procedure. On Tuesday, they go in, they do a craniotomy, they implant the electrodes, and then the patients sit in the hospital room for 1 whole week, 7 days straight, essentially just chilling and waiting to have a seizure so that the clinical team can localize their seizure focus. Okay. The following Tuesday, the surgeon comes back, they remove the electrodes, and after they've localized everything, and they actually perform the resection surgery. Now, what we realize is that during these 7 days, these folks are in the hospital with 100 electrodes in their brain, just chilling, right? Doing whatever it is that they would normally do in their normal lives. They're not being instructed to do much of anything at all. They're doing things like they're talking to their family, they're sleeping a lot, they're playing on their phones and watching TV, they're having conversations with both the clinical team as well as members of their family or friends, whoever comes to visit. They're just living their lives, okay? And we have 24/7 continuous brain recordings and video and audio, and the hospital was throwing it away afterwards. Okay, so that was what the paperwork was about. We did the paperwork so that we can get their consent to just keep their data afterwards. We weren't going to do anything. We didn't ask them to do anything that they wouldn't have done anyway. We did not add anything to the clinical monitoring process. We just got their permission to keep their data. Okay, that's the data set. That's it. All right. I believe the next thing that's going to pop on the screen is a little bloody. So if you don't want to see something bloody, don't just look away for a second. This is what the implant looks like. So the craniotomy is like yayish big, usually on one hemisphere of the brain. They open it up. These are the electrodes that are being put on top. They do a durotomy, right? So then it's directly on the cortical surface. Each of these electrodes is about 1 centimeter center to center. So that's the grid size, right? So it's an 8x8 grid, which means it's like a 7 by 7 centimeters square grid. And here's an X-ray of what it looks like. You can see that there's a couple of different sizes that they use. We, as a research team, make no decisions with respect to what electrodes they use and where they put them. That's entirely up to the clinical team. They put them wherever they want them. We just get all the data afterwards and we can reconstruct stuff. That make sense? Okay, any questions about the data set before I keep going? Okay, I will emphasize here because everyone always asks me, why don't you do EEG in everything that I'm about to describe to you? The short answer is we can, and we actually have, and there's actually ongoing work right now where we're doing that. But especially back when we started, because it wasn't really going to analyze this type of naturalistic data set before, it was too messy, like at least in the beginning. So EEG is great because it's not invasive. But here I'm showing you a picture from our friend's lab where they did simultaneous EEG and intracranial ECOG, right? These large reflections here, these are not signals. This is like they move their head a tiny bit, okay? So the amount of artifacts that was present in the also EEG recording data set that we had access to was just like kind of overwhelming, and we decided that we're just going to work on the ECOG for a little while because the data is cleaner and we were trying to figure out what to do with this gigantic data set, and we didn't need to also contend with motion artifacts. Yes.
We have a question from the Slack channel.
Great.
Basically, do you think there's any hope for decoding things like speech and arm movements from non-invasive EEG?
Speech, no. Arm movements, yes, but it depends on the resolution. I'll show you that we can decode arm movements. Hand movements, probably not. It's a resolution issue, as you may expect, right? I just think like the signal is just not able to go through that many layers of cerebral spinal fluid and scalp and bone and hair. It's just like it's all muddled. Yeah, it's a resolution issue. So no, I don't think we can get speech. I'm skeptical anyway. Gosh, it's hard to say never, right? Like, can you do it if you slap an LLM model on top of it?
That's kind of what they're asking. I'm not optimistic.
Like here's my risk tolerance for something like that. I'm not optimistic enough that I would put a graduate student on it because I don't think it's like we'd had to have a plan B if that didn't work out, right? Like that's my risk tolerance. Do I think it's categorically impossible? Like nobody will ever do it? No, because I don't think it's physically impossible. It's not like traveling faster than the speed of light, impossible, right? Yeah. Okay, cool. So what we got is this giant data set. We have a lot of patients so far. They're all continuously monitored for 7 days each. The electrophysiological data is usually about 100 electrodes, random locations, wherever they wanted to put them. Sampled at 1 kilohertz. We have video, it's RGB during the day. It's infrared at night. And we have audio monitoring in stereo. We'll emphasize that the video camera is a clinical video camera, so the resolution is not great. We fought for a while, try to get a new one in, but fighting with the hospital was not a great idea, so we gave up. The nurses will often come in and actually move the cameras, and so they're not in a reliable position either. So sometimes they'll move around, they'll change the zoom and stuff like that. Whatever they want to do, we just deal with it, right? Like they make the decisions, we deal with the data afterwards. Okay, that's the data set. Oh yeah, and so like I said, all of this is started by our first graduate student, Nancy, and every single thing I'm talking to you about is in collaboration with my colleague and close collaborator, Rajesh Rao, who's in computer science. Right, Raj and I have co-advised a series of students and postdocs in the many year sense. So everything's done with Raj. Cool. All right, cool. So the first thing we decided to do was, okay, so we had this data set, but you know, like analyzing raw video is like really, really hard, right? And so the first thing we decided to do was we wanted to annotate some behaviors from the raw video. And the easiest thing that we could think of to do was tracking their upper limb poses. So their patients are usually sitting in a bed and they often have a blanket up until their waist, so we couldn't see their legs anyway. And it's not like they're walking around playing tennis. So they're mostly moving the arms. So we used, we actually developed like over the years, a series of computer vision tools to track their upper limbs. But you know, every couple of years we kind of try to make it a little bit better. The first data set that we published was in 2017 and had 4 patients tracked. The new one we published a couple of years ago. We call it the Agile 12 data set, Agile for Annotated Joint. I don't remember what it stood for. It's behind that thing. It was cute at the time. So the new data set is the Agile 12 data set, which we published a couple of years ago. I'll tell you about that data set later because we spent a bunch of time curating it because we felt like we had only mined such a small fraction of what was in that data set. We wanted to share it with anyone else who wanted to use it. So it's in NWB format, Neurodata Without Borders. It's on the Dandy archive. And there's actually a little dashboard that goes with it. People have used it for projects, like other research papers have been published using that data set. And lots of students have tried using it for projects, including at like hackathons and stuff. So the NWB hackathon often has people trying to use it. I know this year, NeuroHackademy has a couple of projects that are using our data set as well. So it's like in a package, relatively accessible format for how big it is. Okay, so that data set is actually now out there. So the data set was the most recent version of the data set was mostly curated by two people who joined the last sentence, Nancy Satpreet Singh, who's a graduate student, and then Steve Peterson. You'll notice that they're both at Meta. There's a reason for this. Actually, I think Scott took up a postdoc. I think he quit his job at Meta. He's a postdoc now at Baylor. Anyway, so we have this data set that has a lot of information about these naturalistic upper limb movements. And so I'm just going to show you a small subset of what we've got here. So this is just an example of what we got from the data set. What we've done here is after we track the upper limb movements of these individuals, we looked for instances where they did relatively repeatable things. That's the power of this data set, right? There's no instructive movements, but since we have days and days and days and days of it, we just look for coincidentally when they kind of did a similar thing, but repeatedly on different days at different times. So here's one individual, their face is blanked out, but they've given us permission, written permission to share their video and audio for research purposes, as long as it's de-identified. So this individual, all of these little videos are synchronized for the initiation of movement of their right hand. So if you watch it over and over, you'll see that they're repeatedly moving their right arm. So like kind of line them up so that they're starting to move their right arm. You see that? Okay. So I'll point out a couple of things here. And we've just going to giant pile of these for a bunch of people, right? But in these 16, you can already see that there are some things that point out the power, but also the challenge of analyzing this data set, right? We can synchronize it so that we're getting the initiation of this right arm movement. That's fine, right? You can see that they're not always doing the same thing. Sometimes they're reaching up to touch their head. Sometimes they're only moving a tiny bit and putting their arm back down. Sometimes they're like reaching out to eat something. That's often why they're moving their arms, is for reaching out for a drink or yogurt or whatever it is, right? There's also challenges with lighting. So some of these are videos at night. Some of them are videos during the day. Right. So those of you who may have worked with computer vision in some way, you know that the lighting is like makes things really challenging. And so even in Seattle, the sun does sometimes come out and like, they open the window, the lighting totally changes and it makes tracking really difficult. Another thing is that there's often lots of occlusions, right? There's not often a clear view of what their hands actually doing. Sometimes they're doing the uni arm movement. Sometimes they're moving both of their arms at the same time. Sometimes they're alone in a room, at least as far as we can see. Sometimes there's another person in the room, right? Like, you know, like there's a person right here doing something and interacting with them. I'm not showing you the audio here, but of course we have the audio signal. And so sometimes there's silence and nobody is talking. Sometimes they're moving their arm and talking at the same time, or listening to somebody talking to them. Okay, these are all the variety of different behavioral variables that modulate the underlying phenomenon of initiation of the right arm movement that we were trying to study. So the first thing we actually did before we get to the neural activity is just quantify the variety of movements, which actually had never been done before. So out of these 12 people, we looked at a bunch of metadata of the variety of their movements. And you'll see that they're like largely consistent, but lots of people move differently. This is not surprising to you. We can also characterize a bunch of other things, like how often they're moving and speaking, as well as how often they're moving their one arm versus the other arm. So a bunch of these metrics of this kind. Now, we went back and then looked at the electrocorticography to look at what are the neural signatures of this initiation arm movement, right? For us, nobody, like tons of people have studied this. This is well studied in the experimental context. Graduate student goes in and says, please move your arms, they move the arm, please move your arm again, they do it 10 times, right? And you average over that, you get a beautiful signal, but no one's ever looked at it in a naturalistic context, right? What does your brain look like when you're moving your arm without a graduate student telling you to move your arm? Okay, so this is what it looks like. What we did was that we summarized the 12 subjects, the 12 participants we had, projecting everyone onto a common brain atlas, NeuroHackademy. And then we aggregated the data sets onto a couple of parcels of the NeuroHackademy brain. So here are the parcellations. And if you look over the aggregate information, the power spectral features associated with the initiation of movement is very consistent with what we expected from experiments. Okay. In particular, if you line up all of the movements at time 0, so -1 is right before they moved, +1 is right after they moved, you'll see that there's this very stereotypical pattern right here, especially in the precentral and the postcentral regions of the brain, right in front of and behind the central sulcus, where sensory motor cortex is. That's exactly what we expected. In particular, we see that there's an increasing power in this high frequency band and then decreasing power in this low frequency band. This is exactly what people have reported before, including former people that Raj have worked with. If you look at temporal cortex, there's almost nothing, no movement associated changes in spectral power. So far, so good, right? Okay, so far, so good. Everything totally makes sense. We haven't discovered anything totally shocking and new. This is good. This basically these things are working, okay? And then we looked at every individual. And my goodness, it turns out that there's a huge difference between my brain and yours, right? And if you are a data scientist or a statistician, you might be like, oh, that's just like noise and variability. But that's not noise and variability, right? Like the difference between my brain and Noah's brain is the difference between me and him. And I value that difference. Okay, so here's the difference between these 12 individuals we looked at and what their power spectral densities look like if you just looked at them one at a time by themselves. Now, you'll see when we fall that there are some people here where they look very much like what we expect out of the average brain, right? Like S3 here has this high frequency power, like there's an increase here and a dark band down here, so far so good. But there's people who don't look like that at all, like S8 has the increase but not the decrease, right? You have people where there's a decrease here, but not really an increase. S1 has the increase and the decrease, but the frequency band of the decrease is significantly wider than it was on average. And so this also speaks to the fact where if we were trying to decode movement from this type of data, right, you're going to have to really tune your model, see each individual. And this is why people have had to tune their models individuals for so long, because this is the extent of the variability, even for a relatively simple thing to decode, like moving your arm. Is there a question? Yes.
Kind of two. One is that the placement of the ECOG, was that variable? at all?
Yes. Yeah.
So that-
Quite. Yeah. We had to do a bunch of work with projecting it onto the common brain, and there were assumptions made. Yes, because we had no control of the placement.
Do you have any information about where the placement was? Like,
Yes, we know precisely where they are. Yeah, we have the structural MRI as well as the X-ray, and we triangulated everything. So we have 3D coordinates where every single electrode was. Yes. That's all unpublished data set. If you're interested, you can download it from the internet today. There was a question in the front. Yes.
So this is average per participant for a few days, right?
Yeah.
So could it be, for example, due to inter-individual differences, could they be coming just because, for example, some people tend to move their arms at the same time, so they would not see the sequence and because they would actually.
We looked at a bunch of the metadata parameters, and there was not a particularly great pattern that we found. We did all kinds of things. We looked at time of day. We looked at whether or not it's one arm or it's two arms. We looked at whether or not it's when they're talking and moving versus just moving. There were some small correlations, which we put in the paper, but they were not so huge that I would like to make a big deal about it. We did look. Great question. Yes.
Are these for particular
Say it again, please.
these different subjects? Is this for particular?
Is this for particular? Oh, yes, sorry, I forgot to tell you. So all of these is for the precentral region. We have these for all of the other ones, but I'm just showing you the one for which the pattern is the biggest.
Is the case that not everybody has the same signal? Or is it the case that everybody has the same signal but in a different way?
So it's that not everyone has the same signal. Yeah, like if you look this is consistently where the largest patterns are. And so it's not like this person had it in temporal, like that's not where it is. Did you have a question?
Yeah, I guess I was gonna ask something similar, which is
Yeah.
how much of this is true individual variability and how much of this is measurement variability. I imagine you have different numbers of trials per subject.
Yep.
and The electrodes are positioned in slightly different angles and things of that sort. So how much of this do you believe is really, if you had measured, you had like full control of the measurement and you measured fixed number of trials, do you think you'd still see this
Yeah.
level? Yes, okay.
My belief is that we will. The thing that we can't control, obviously, is that everyone's brains are different. So I've looked at a bunch of these MRIs where I was scratching my head going like, where is the central sulcus? It's like this if you look at a textbook, an undergraduate textbook, there's like a beautiful cartoon of the brain, and there's very obviously a central sulcus right down the middle, right? And there's the motor cortex and the sensory motor cortex. And I'm like, okay, this is what human brains look like, right? And then we go look at the clinical data, and there was a couple of people where I'm scratching my head going like, where's the central sulcus? I don't know where it is. I don't see it. And I thought it was because of me, because I'm not like trained as a human neuroanatomist, right? So we go and ask our neurosurgeons, and apparently they were like, oh, I don't know. Sometimes there's no central sulcus. It's like, I didn't realize that that's the extent of the variability in the anatomy of human brains, that like there'll be individuals out there, maybe some of you, I don't know if you've had MRIs, where it's not obvious where your central sulcus is. Like I didn't know that could happen. And so, yes, like I don't know how to account for that, right? Here we're projecting it onto functional space, so to speak, but we're making a lot of assumptions about anatomy. And so I don't know how to account for both the anatomical variability as well as functional. So we're doing our best. That's all I got. But I do believe that the variability is actually there, not merely as a consequence of our inability to line people up. Yes.
With people with weaker signals or like more variable signals, did you find that was the clinical area where they like later had surgery at, or is it like not related to that?
Great question. The vast majority of the focus of their seizures was in temporal cortex. So like some of these regions down here. In fact, we had to like dig pretty hard for this is why we have 12 of these people instead of like the dozens and dozens that we actually have data for, is because it was like the surgeon tended not to put the electrode array up here, where sensory motor cortex of the arm and hands are, because that's sort of not where their seizures tended to originate from. And so I think we are reasonably staying away from the neuroatypical part of these brains. Yeah, good question.
Hello, sorry, I have a couple of questions as well.
You're listening.
Yeah, thank you. So the first question is, what kind of analysis did you do here? It was like a time frequency analysis, and the event was when the person was like moving the right hand, or like how was it? That's my first question. You can maybe answer this one first, and then I ask the other one. So that.
Yeah, sure. Yes, that you're exactly right. Yeah, we looked at all the instances where they're moving the right arm. That's time 0. We took a spectrogram of every event and then averaged them. Yep.
Okay. And then the second question, it's how similar were the participants in the characteristics? For example, age. I know, well, maybe
Not very. They were everywhere. They're all adults because we did not have a pediatric data set. But they were anywhere from like early 20s to 50s and 60s. The demographic details are in the paper. I don't recall exactly what they are, but it was quite wide. I mean, we had both male and female, and people of different ethnicities and demographic characteristics as well. So yeah, not similar to each other.
Yeah, I was asking because maybe, of course, that introduces a lot of variability. I was asking because
Yes.
I do EEG with older adults, and old adults as well. They have a lot of variability because of the same things, right? Like I think that when you are 20 years old, for example, you have gone through a lot of different things, but not as many as when you have been 60. You know what I mean, or 70 or 80. So I wanted to ask you, what do you do in these cases where you have like not a large sample and a lot of variability due to the nature of the conditions in your case, for example, that you cannot pick the participants that you would like to, or my case, for example, where we have old adults and they're very, very different between them?
Yeah, so in this case, I'm taking more of an my perspective on this is more of an engineering approach. which is that I'm not interested, I mean, like, I am interested in the variability because I think is really interesting, but given the constraints of this very precious clinical data set, there's only so much we can do. So I'm focusing on asking the question that we can ask rather than asking questions that we really have no control over. Like, you know, ideally you would have a few thousand people and control for everything. We can't do that. Like, we literally can't, right? So I'm more focused on the engineering question that drove us in the first place, which is that these are all people who may want to break a computer interface. Like, we don't control anything at all, right? Like, a new person could walk in with whatever variability they have, whatever age they are, and say, I may need a device, right? So how do we just use the data we have to make it so that we can train a decoder for them as well as we possibly can, right? And so that's what kind of what we set up to do. I understand I'm at the very end of my time, so I'll just stop talking in like 2 minutes. But what we ended up doing was developing a set of decoders that are able to do what we call transfer learning. So we trained decoders to detect the initiation of movement of this right arm. And by aggregating all the data that we had, rather than training bespoke decoders for every individual, we trained it for like 11 of the people we had and then tested it on the 12th person. So we're doing this cross-validating thing, right? And by doing it that way, what we're able to show is that our decoding architecture can actually transfer to a new person sight unseen, as if they had walked into the clinic next month, right? And I'll tell you the details about, and this is all published stuff. So you can read about it if you want to. It's not like there's nothing secret about any of this. But what we were able to accomplish was build generalized decoders. So within the same modality means ECOG to ECOG. So we're training it on 11 people, testing it on the 12th person. And then with a tiny bit of fine tuning on the 12th person. So using a small handful of trials from that particular person, we were able to do even better. And then kind of impressively, I mean, when Steve, my postdoc Steve did this, I was like, there's no way it's gonna work. I was like, I'm gonna try it anyway. And then he tried it and of course it worked, so he was right. We trained it on ECOG people and then used another data set that we actually got from the literature. We downloaded somebody else's EEG movement decoding data set. And we were able to transfer from the ECOG decoder to the EEG data set. The opposite did not work, by the way. We trained on the EEG and never worked on the ECOG. And so fine tuning worked a little bit better there as well. So there's this whole line of work where, again, this is not, we haven't tested this on any patients. This is not an experimental study. This is just like a concept piece, right? This is saying that if in the future we might like decoders to work on people sight unseen, what would we do? And conceptually, our idea is that you would use the wealth of data that we have already collected in the past, counting on that our brains are approximately like each other. So we train generalizable decoders that kind of work for most people. And then with much less training data on every individual, we can get it to, we can tune it up so that it's individualized to you. But instead of starting from scratch, we start from a model that generally works for lots of people and go from there. Okay, that's it. I'll stop talking. There's more, but I don't care. Yeah.
How prototypical or atypical was this 12 person that was tested?
We did every permutation.
Oh.
So everyone took a turn being the 12th person and the results are averaged. Yeah. Good question. Yeah.
So might the model be like capitalizing on different clusters of phenotypes associated with the different patterns?
Great question. We in our 12, which again is like a small number in the grand scheme of things, right? We did not observe clusters. It was really a continuum. So there might be clusters, but you know, I don't know. We haven't seen it. Yeah.
What did you guys think about the previous slide when we were trying to compare?
So I think of it as I'm going to say this in kind of like mathematical language. If that, I don't know if that makes sense. You're going to do it anyway. So when you're modeling something in the physical sciences of math, usually there's like a 0th order approximation, right? And then it's going to be wrong, and you make a first order correction, and that's also going to be wrong, and then you add a second order correction. Right. So it's not like the first order model was wrong. It's just like it got you like kind of in the right area, right? And you make a correction, it's like, okay, now I have the right slope. And then you add another term like now I have the curvature. Right. It's more like that. It's not like everyone is just completely different. Our brains are similar, right? So getting like the 0th order model, it's just getting you in the right neighborhood, and then you do a correction for maybe like some aspect of variability. And then you add another correction that makes it so that it works for that individual, like really, really well, right? So I don't think it's like none of those models are wrong. It's just a matter of how much data you have and how close you want it to be. Right? Does that analogy make sense to you? I don't know if you're a physicist or like, okay. So the other analogy that I have for this is so like we all know from classical mechanics that if you drop two objects, regardless of their weight and mass, they're supposed to fall to the ground. Right? Okay, this is great. Like, it's not like that's wrong or anything. If you took a feather and a bowling ball to the Tower of Pisa and you drop them, I guarantee one of them is going to hit the ground first. So if you did an experiment, you might think to yourself, wow, F equals ma is wrong. But that would be the wrong conclusion. It's not wrong. It's just that that's the first order approximation, on top of which we have to add another correction term for air friction. Right? Both of those things are true. So we're kind of building the average approximate model, which I believe to be not wrong, but it doesn't work well enough depending on how accurate you want your prediction to be, right? So if you actually want to know when the falling ball is going to hit the ground and when the feather is going to hit the ground, you need a better model. But it's not like F equals ma is wrong.
Do you have any idea what
It's hard to say. Great question. I mean, like we are digging into it. And coming up with more practical strategies of dealing with that, right? So at that level, it becomes less a matter of describing the data and becomes more of like an engineering application, right? For a task you want to accomplish, there is usually a margin of how well you have to do for this tool to be useful, right? Same thing with self-driving cars. Self-driving cars don't have to be perfect, but there's a safety margin, right? Like we need these cars to be this safe so that we feel okay about having them on the road. Right? We need our decoders to do this well, so that in this context, they're useful for us. Like we can use it to help people in some concrete way, in this way that's going to fit into our workflow of how we're going to help these people, right? So that's the design criteria. We need decoders for this type of data on this type of task to work this well. And that number is not 100%. It's also not as much as possible. There is such a thing as good enough. And so for every task, we're trying to build a decoder that is good enough to fit into the pipeline of who it's for and how we're going to use it. Yep.
So at the beginning you mentioned that sometimes there is ECOG and EEG,
Yeah.
but that is not the case for your data sets. It was just an example for
That was just an example. We don't have simultaneously collected EEG and ECOG. I don't think that's typically done, which is why I dug up that paper. It was one of the few I've seen where it's simultaneously fired. Yeah.
Because I was thinking that maybe in that case that you can disentangle if you train within subject and then kind of do all the processing across subjects.
Yeah. If you had simultaneously reported data, yes, that would be a thing that we would do. Thank you.